Hi everyone, my name's Emily and I'm going to talk to you about something called environmental DNA today, or eDNA for short. What it is, how it's used, some examples, and pros and cons. So let's get started. So what eDNA is, it's basically DNA that's extracted from an environmental source. So what that would be is something like the soil, air, water, could either even be biological, so like seeds or hair of an animal, glaciers, sediment is kind of commonly what I've seen. It's the samples have been taken from. So those samples could either be current, so recently taken, or some of the samples like from the sediment of a lake or a stream might get you DNA that's like ancient, maybe even millions of years old. So a few different ways or reasons for looking at the DNA. What you do with that sample then is you do a PCR, which is polymerase chain reaction. And basically you're looking at the sequence of the DNA to help identify what that DNA is from. So they do something called DNA barcoding or metabarcoding, and they're looking at like certain markers on the DNA to say, that this is this kind of plant or this is this kind of animal. So they're trying to figure out what's in the sample or what's been in that sample to figure out kind of the makeup of that community. So what plants are there, what animals are there, what microbes, and all that good stuff. So really interesting how they do that. This is a really new thing. They just kind of started doing this in the 2000s. So when, our DNA technology started getting a little bit better, this has become a newer thing and even bigger and bigger as the years go on. Okay. So again, kind of taking samples either from water, air, soil, or from a biological source. And it's pretty streamlined at this point, I guess, how they get the samples. So they're taking these little core samples. It could even be from snow and things like that, kind of like they do ice samples for climate change studies and things like that. So it's kind of similar to that. Then you take that DNA, you bring it into the lab, and you break it down and kind of isolate the DNA and look at the specific sequences to find what species you have. So there's a lot of different applications with this. So here, let me move this over a little bit so you can see. So a few things you can do. You can look at like evolution. So if you're looking at like an older sample, you can look at ancient ecosystems, how they've changed over time. Pollinator interactions, and I'll talk about an example of that in a minute. So you can actually look at DNA within like the honey of a bee and figure out, what that bee has been using to pollinate. So that's kind of interesting. A diet analysis, so you can look at what animal and plant DNA is kind of together and see what resources each species might be using. Invasive species detection, so again, looking maybe over time how invasive species have changed in certain areas. Pollution response, air quality monitoring, so also for like a human impact analysis, how has the composition of an area changed before or after human impacts and things like that. So a lot of interesting, really cool things you can do with eDNA. A few specific examples that I wanted to share. The first one is what I kind of just mentioned. So scientists in a study, they looked at what plant DNA was present in the honey samples of certain bees to determine which plants those pollinators were using. And this is going to be really important because, as many of you may know, our pollinator populations are declining, like very rapidly. The bee populations are declining, and everyone's kind of like in a panic about it. What do we do? And so if we know what plants the bees and all the other pollinators use most, that can really help with conservation efforts. And, as we need food crops, we need those pollinators for our food crops. This can help us kind of get more informed decisions about that kind of stuff. Another one that I thought was pretty cool was a rare carnivore species study. So the Canada lynx, the fisher, and the wolverine were being surveyed to just better understand their population numbers, because it's a really rare and elusive species. And they were actually using eDNA data from the snow samples from like the tracks that they saw in the snow, and they were doing the analysis of that to get a better idea of how many of those kind of rare carnivores were in that population. So I thought that was really cool. Another way to kind of find more elusive animals that you don't usually see when you're doing like an in-person count. So that was kind of some examples. I found a lot of pros and cons. So instead of just highlighting a few, I wanted to show you all of, a lot of the ones that I saw. So some of the main things that I think are going to be important here is way down here at the bottom, cost effective. So every study I looked at, they always like, oh, this is really cost effective. Since we're getting so much better with PCR and our lab analysis, seems to be like an easy way to do this. It's non-invasive, which I also like. So you're not having to like tag animals to count them. So that's a really big pro, I think. And then let's see. So better understanding, jumping back up to the top, it's going to help us better understand microbial diversity. So that's something that we don't have as much information about as like say mammals. So this would be a good way to kind of figure out what our numbers look like for that type of species. Better detection and census counts of endangered species helps us understand ecosystems as they were prior to human impacts, which I think is huge in helping us move forward with conservation planning. It can be used to study a wide array of taxonomic groups at a time, so it's not like only certain species will work for this. It's kind of like everything seems to be working for doing this. Under good conditions, eDNA can survive up to 1 million years. So that's pretty amazing that you can get like this time scale of really wide time scale of changes over time. Can help us better understand native versus non-native species. So using longer time scales and core samples. So really understanding, how did things get here? When did they get here for certain invasive species? Can be used to study how species respond to extreme catastrophes, environmental change, and lastly, how standardized the process is. So it's pretty, well documented how to do this in a proactive way. Okay, and my list of cons is much shorter, which is good. So the first con, difficulty with detection of taxa. So sometimes you might not get a good enough sample of DNA to actually identify what species you have, or you don't have the markers on the DNA that help you identify that. PCR primers may miss certain taxa or DNA concentration may be too low for detection. So that's might have like a false positive or a false negative read on those. Ancient DNA has a higher probability of contamination, either within the lab or outside of the lab. You can kind of get a mix of your DNA. That seems to be a common issue. The movement of DNA between soil can lead to a misinterpretation of the spatial and temporal scales of the population. So like, your strata in your lake, for example, can kind of mess up your readings, I guess. And then the DNA can be degraded over time, either by biotic factors such as UV radiation, temperature extremes, or biotic factors such as microbes or enzymes breaking things down, and that's kind of a big con. Future use, so it's kind of the last topic here. Where are we going in the future with this? There's so many different places you can go, so many different things you can do this with. We've only gotten a select few samples, but kind of just better understanding of species abundance and diversity, better understanding how populations and landscapes have changed based on human development and industrialization. And then the last thing, I thought it was really cool. I read this article about how a high school in Denmark is doing citizen science projects with estuary samples and kind of partnering with a local museum to monitor the health of their local environment. And I thought that would be a really awesome thing, for us to do here in the U.S. or really anywhere to monitor, changes over time of your local environment. So hope you learned something, and hope you enjoyed eDNA. It's really awesome and something to look more into as we go forward. So thank you.